Okay, imagine you have this incredibly powerful medicine, something that could really zero in on disease cells.
Hmm.
But here's the catch, right? Delivering it is like trying to hit one specific house, but you're using this enormous cannon.
Okay, yeah.
You definitely get the target house, sure, but you level everything around it too.
Yeah.
The park, the neighbors' places, just collateral damage everywhere.
It's effective, but devastatingly messy. Not ideal.
Not ideal at all.
Yeah.
What if instead of that cannon, you had something more like a surgical sniper rifle, delivering that same power, but only hitting the exact spot you need it to?
That level of precision, I mean, that's the dream, isn't it? Especially for treating things like aggressive cancers. The line between killing the tumor and harming healthy tissue is just so fine.
It's critical. Minimizing that collateral damage, it changes everything for patients. Outcomes, quality of life,
Absolutely.
and that whole quest for precision. That's what really pulled us into some fascinating research coming out of Poland from scientists at Wroclaw Medical University.
Oh, yeah. What are they looking at?
They're exploring this pretty novel way to fight a particularly aggressive childhood cancer. It involves a potent natural compound and, get this, electricity.
Electricity. Okay,
Yeah.
that's interesting.
Yeah, so we've been digging into their work, specifically a paper they published in the journal Bioelectrochemistry. It's called Optimizing Jasplakinolide Delivery in Rhabdomyosarcoma Cells Using Pulsed Electric Fields, PEFs, for Enhanced Therapeutic Impact. Bit of a mouthful.
Jasplakinolide, pulsed electric fields,
Oh, wow.
rhabdomyosarcoma. Okay, lots to unpack there.
Exactly. So that's our mission for this deep dive. We want to understand the challenges, of treating cancers like rhabdomyosarcoma, and then see how this combination, this natural stuff and targeted electrical pulses might just be the key to making that surgical sniper delivery a reality.
That's like a plan. Where do we start?
Let's start with the compound itself. Jasplakinolide, it sounds kind of exotic.
It is actually. It's a natural chemical. And the origin story is pretty neat. It comes from marine sponges found in the Pacific Ocean.
Wow, okay. From a sponge, what does it do?
Its real power lies in what it does inside a cell.
Yeah.
It targets the cytoskeleton.
The cytoskeleton,
Yeah.
like the cell's internal skeleton. It's scaffolding.
Exactly like that. It's made of these things called actin filaments, and it gives the cell structure, lets it move, and critically, it's essential for cells to divide and grow. Jasplakinolide basically messes with the scaffolding. It stabilizes the actin filaments, kind of freezes them.
Freezes the scaffolding so the cell can't function properly, especially cancer cells.
Precisely. Cancer cells are all about uncontrolled growth, division, movement,
Yeah.
how tumors invade and metastasize.
Right.
So disrupting this fundamental cytoskeleton structure really throws a wrench in the works. It can directly lead to tumor cell death.
Okay.
And Dr. Anna Schuzik, one of the researchers on this paper, points out it seems particularly effective in cancers that really rely heavily on that cytoskeleton for being invasive. Things like breast cancer, lung cancer, gliomas, and the one they focused on, rhabdomyosarcoma.
Right. So it hits something fundamental, which makes it powerful. But you mentioned the cannon problem earlier. There must be a downside to something this potent.
Oh, absolutely.
Got it.
That's the core issue. Jasplakinolide is powerful, yes, but it's not smart. It doesn't know the difference between a cancer cell and a healthy cell.
Oh.
So if you just give it systemically, like in an IV drip, it goes everywhere and affects all your cells. That leads to major toxicity.
Okay, so that's the challenge they're tackling. How to use its power, but only where it's needed.
Exactly. Which brings us to their specific target, rhabdomyosarcoma.
Right, RMS. Tell us a bit more about that.
Yeah, RMS. It's thankfully relatively rare, but it's a very aggressive cancer. It starts in immature muscle tissue, the kind that develops into striated muscles.
And it mainly affects kids.
Yes, primarily young children, often between the ages of 1 and 10. It can show up in various places: head, neck, genital area, arms or legs are common. And sadly, it accounts for about half of all soft tissue sarcomas in that age group.
And treating kids, standard chemo and radiation, they work, but the side effects on a growing body can be really harsh long-term effects too.
That's exactly it. The long-term impact can be profound. That's why finding more targeted therapies is so crucial, especially in pediatrics. The Wroclaw team, led by Dr. Schuzik, they saw the potential in jasplakinolide for RMS, but knew they needed a way to contain its effects.
And that's where the electricity comes in. Electroporation, you said.
Correct. Electroporation, or sometimes called PEF, pulse electric fields. It's a technique where they apply very short, controlled pulses of electricity to the cells or tissue.
Okay, short bursts of electricity. What does that actually do to the cells?
Well, think of the cell membrane, the outer wall. These pulses cause tiny temporary pores or openings to form in that membrane. It's like quickly opening a door that's usually shut tight.
Just for a moment. Long enough for something to get through.
Exactly. Just long enough for molecules that normally can't cross that barrier easily, like, say, a drug molecule to slip inside the cell. Then, almost immediately, the membrane seals back up.
Okay, that makes sense. Why is PEF a good candidate for this kind of delivery?
Well, the researchers highlighted a few key advantages. First, it's not some totally new experimental idea in the clinic.
Right.
Professor Jelita Kobaka, another researcher involved, notes that PEF is actually one of the most clinically advanced targeted delivery methods being looked at. There are approved devices, electrodes. They've been using a related technique, electrochemotherapy, for over 20 years.
So there's experience with it. It's not starting from absolute zero.
Right. It has a track record. And here's a really crucial part for targeting. Healthy cells seem to be much less sensitive to these electrical pulses compared to cancer cells.
Oh, really? Why is that?
The exact reasons are complex, but cancer cells often have different membrane properties. This difference in sensitivity is a huge built-in advantage if you're trying to be precise.
That is a big deal.
And another point from Wojciech Slasa, also on the team, is its versatility. You can use electroporation to get various things into cells, not just standard chemo drugs, but potentially immune-boosting molecules, or even components for gene therapies.
So it opens doors, literally and figuratively, for different kinds of treatments, maybe even combinations.
Exactly. It seems like a really logical partner for something potent but potentially toxic, like Jasplakinolide, a way to guide it.
Okay, so let's get into how the Wroclaw team actually tested this combination in their study.
Right, so they did their experiments in vitro, meaning in lab dishes, not in animals or people yet. They used the target rhabdomyosarcoma cells, the RMS line, but importantly, they also included two types of healthy cells as control hamster ovary cells and mouse muscle cells. This lets you directly compare the effects.
Good comparison. So what happened when they hit the cells with both the jasplakinolide and the electric pulses?
The results were, well, pretty striking. They observed a dramatic drop in the survival rate of the RMS cancer cells, a huge reduction.
Okay, good for the cancer cells. What about the healthy ones?
That's the key part. That same combination, the drug plus the electricity, had almost no significant effect on the viability of the healthy hamster or mouse cells.
Wow. So it really did act like that sniper rifle. Hit the target, spared the bystanders.
That's exactly what it looked like in the dish. And they dug deeper, looking under the microscope at what was happening inside the cells. They saw clear signs of the cytoskeleton, that internal scaffolding we talked about, basically disintegrating or collapsing, but specifically within the cancer cells treated with the combo. Dr. Chusik described it as the internal structure just falling apart.
So the drug, guided by the electricity, was effectively dismantling the cancer cell from the inside out.
That seems to be the mechanism, yes.
Yeah.
They also looked at another protein, pancadherin. It's involved in helping cells stick together.
Cell adhesion. Important for tumor structure and spread.
Very important. They saw changes suggesting this protein was fragmenting in the treated cancer cells. This implies the cells might be losing their connections to each other, which, theoretically, could make it harder for a tumor to hold together or for cells to break off and metastasize.
Okay, so really promising results on multiple fronts, but we have to stress, this is in the lab for now.
Absolutely crucial point. This is in vitro research. It's a fantastic proof of concept, but it's not a treatment ready for patients tomorrow.
What are the big next steps then?
The standard path is, first, testing in animal models. You need to see if this works in a whole complex living system, not just cells in a dish. How does it distribute? Are there unexpected side effects?
Makes sense.
And then if those studies look good, the next stage would be carefully designed clinical trials in humans, starting probably with small safety studies.
And moving this into clinical trials, especially for children with RMS, what are the big hurdles they anticipate?
Dr. Anna Shusik talked about this. The human body is just vastly more complicated than lab models. You've got the tumor microenvironment, all the other cells and structures around the tumor. You've got variations in tissue conductivity, which affects the electricity part.
Right.
Patient anatomy differs. Children metabolize drugs differently than adults. There's the need for general anesthesia for the procedure itself. It adds layers of complexity.
And RMS being rare doesn't help with organizing big trials either, I imagine.
Exactly. Recruiting enough patients for large-scale trials needed for drug approval is always harder with rare diseases.
So moving to more complex lab models first, like the 3D tumor spheroids Wojciech Slasa mentioned, that seems like a vital intermediate step.
Definitely. These 3D models, they better mimic the structure and environment of a real tumor. It helps bridge that gap between the petri dish and a living organism. It's a key part of the preclinical work.
What about other future directions this research might lead to?
Well, one really exciting avenue is combining electroporation not just with drugs, but with immunotherapy.
Ah, using the body's own immune system.
Right. The idea is to use the PEF pulses to deliver immune-stimulating agents, things like cytokines or checkpoint inhibitors, directly into the tumor area.
So you'd be activating an immune response right where you need it.
Potentially, yes. Delivering it locally could ramp up the anti-cancer immune attack within the tumor while minimizing the often harsh systemic side effects you can get when these agents circulate throughout the body.
That sounds very promising. Professor Kulbaka also had some thoughts on where this whole field of bioelectrics in cancer might go in the next, say, 10 years.
Yeah, she painted a picture of really personalized precision medicine. Not just using one technique, but maybe combining electrochemotherapy, perhaps irreversible electroporation. It aims to kill cells directly with electricity, maybe even plasma ablation.
Yeah, a whole toolkit.
Exactly. Tailoring the specific electrical pulses, the drug doses, adding in local immune boosters, all customized for that individual patient and their specific tumor.
So treatment becomes truly bespoke, not one size fits all.
That's the vision. She also mentioned developing much finer, more precise electrodes to reach tumors that are currently difficult or impossible to treat with surgery or radiation because of their location.
Getting into tricky spots.
And miniaturization, too, could some of these procedures eventually become less invasive, maybe even outpatient therapies. Plus, looking further ahead, integrating AI.
AI, how would that fit in?
Imagine real-time monitoring during the procedure using biosensors. AI or machine learning algorithms could analyze that data instantly and suggest adjustments to the electric field parameters, tweaking things on the fly to maximize the effect on the tumor while minimizing damage to healthy tissue.
Real-time optimization.
Yeah.
The potential benefits for patients thinking about all these advancements seem enormous.
They really do. You're talking greater precision, potentially higher effectiveness by combining approaches, less invasive procedures, true personalization, and maybe even harnessing the immune system more effectively. It all points towards better outcomes.
And let's circle back to why this work from Wroclaw Medical University is so important now, especially thinking about pediatric cancer.
Well, like we said, children's bodies are still developing. They are particularly vulnerable to the toxic effects of standard cancer treatments.
The long-term consequences can shatter them for life.
Exactly. Finding therapies that are just as effective, or maybe even more effective, but are far less damaging systemically, that's the holy grail in pediatric oncology.
So approaches like this Jasplakinolide PEF combination, even though it's early days, offer that hope. A future where maybe we rely less on harsh systemic chemo.
That's the hope, yes. Potentially increasing cure rates for tough cancers like RMS, but doing it with significantly fewer debilitating side effects. That translates to a much better, healthier future for young cancer survivors.
Cancer is still such a huge challenge, obviously, but it's research like this, crossing disciplines, thinking creatively, really focusing on that precision that offers genuine steps forward.
It really is about moving from that blunt force approach towards something much more targeted and intelligent.
Yeah. This specific work, combining the sponge compound and the electric pulses, it really embodies that potential surgeon and sniper idea we started with. Dismantling the cancer cell precisely, leaving the healthy neighbors alone. It's still lab work, but the concept is powerful.
It definitely makes you think.
It does, and it makes you wonder, doesn't it, how breakthroughs often come from combining things you wouldn't necessarily think go together, a sea sponge chemical and electricity. What other unexpected combinations, what other intersections between seemingly unrelated fields might hold the keys to tackling the diseases that still challenge us today?